Hello, and thank you for joining us today. I'm Katie Gillis, Product Manager for the Luminex Guava Microcapillary Flow Cytometry portfolio. I will be joined today by Stephanie Brunelle, the Product Manager for the CellStream Flow Cytometers, as well as Brian Hall, the Product Manager for our Imaging Flow Cytometry platforms. We're excited to review some applications for cell biology using the Luminex Flow Cytometry technology. Luminex offers a variety of flow cytometers for cell biology needs, ranging from our simplified, easy-to-use MuCell analyzer to the Amnis Imaging Flow Cytometers that combined imaging with traditional flow cytometry. Today, we will be walking you through applications for each of our cytometers focused in cell biology. First up, we'll be talking about the Guava Muse and Guava EasyCyte systems that have many applications for cell biology, ranging from simple count and viability measurements to more complex population identification, as well as mechanism of action studies. The Guava Muse and EasyCyte instruments use a patented microcapillary design to provide simple and reliable results. The microcapillary eliminates the need for sheath fluid on our systems. We pull the sample through the microcapillary into the positive displacement pump that allows us to know exactly how much volume has been acquired, which further allows us to determine the absolute counts for each sample without the use of reference beads. Additionally, the microcapillary technology allows for small sample sizes, which preserves your precious cellular samples and produces low biohazardous waste. One assay we've developed and optimized for cellular biology needs is our Guava ViaCount assay that measures cellular counts and viability measurements for your cellular samples. Our simplified, easy-to-use workflow allows you to simply take our ViaCount reagent, add your cellular sample, incubate for 5 minutes at room temperature, and acquire on the EasyCyte or Muse systems. The proprietary ViaCount reagent contains 2 DNA intercalating dyes. One that is membrane permeant, that will stain any cell that has a nucleus, allowing for discrimination between nucleated cells and debris, as well as one that is membrane impermeant, that will distinguish between dead and dying cells. Additionally, we've developed a software platform for simple analysis and acquisition of your samples, with the predetermined plots allowing for easy to identify populations, as well as statistics that have been optimized to highlight the key features of this assay. As the graph shows on the lower left corner of this slide, we've compared the cellular concentration versus the predicate method. You can see that there is linear correspondence with an R-squared value and slope close to 1. Additionally, when we compared the viability measurements using the ViaCount assay to a predicate method, as shown by the bar graph, similar results were obtained. Lastly, one key feature is the fact that our assay distinguishes dead and dying cells, unlike traditional trypan blue methods, which highlight just live and dead cells. So shown here is an example with healthy cells, as well as overgrown cells. As you can see, the overgrown dot plot shows a dead and dying population in the middle here. That sample was then compared to an annexin-5 assay to compare population percentages. As you can see, the population percentages for the annexin-5 assay matched those shown with the ViaCount assay. The Muse and EasyCyte technologies provide pre-validated kits that allow for the identification and enumeration of both whole blood and PBMCs. These assays are mix and read and take as little as 10 microliters of sample. The sample is mixed with one of our pre-formulated kits and acquired on our Muse or EasyCyte instruments. Each instrument has a predefined acquisition and analysis template that allows the user for simplified acquisition and analysis. The Guava counting assays, highlighted here, utilize our proprietary mix of dyes to exclude cellular debris and identify the count of nucleated as well as debris. We also identify viable, dead, and dying cells. Unlike assays that utilize trypan blue, the Guava counting assay uses a DNA intercalating dye that indicates any membrane disruption and can be an early indication of cellular distress. The Guava immunology assays are developed to provide the user with CD4, CD8, or B cell concentrations and the percentages in the overall lymphocyte population. These are also mix and read assays that can be combined to determine all of the various populations. Results for the Muse immunology assays are shown here. The Muse immunology assays provide absolute counts, total lymphocyte counts, and population percentages based on the total lymphocyte population for either CD4, CD8, or B cells. Two of the assays are highlighted here. The dot plots in the upper left highlight the results for either the CD4 assay or the CD8 assay. The lymphocyte population is shown on the y-axis for both plots, and either the CD4 or the CD8 population is shown in the x-axis. Very clear separation for the lymphocyte populations is shown for both assays, as well as clear separation for the CD4 as well as the CD8 populations. A comparison of the CD4 counts for a predicate method versus the Muse cell analyzer is shown in the graph on the right. You can see that based on the linear response that is shown, based on both the slope as well as the R-squared value, that comparable results are seen on the Muse cell analyzer. Lastly, we evaluated the CV results for the count, the percent lymphocyte, as well as the count for the total population. These results were compared for 3 operators across 3 instruments running 4 samples in triplicate. You can see that the CVs for all of these populations were well below 5%. In addition to being able to count your cell samples on the Guava EasySite and Muse systems, we've developed a few assays to assess cellular health. 3 of those assays are the Guava Nexin, MitoDamage, and Cell Cycle assays. The Guava Nexin assay combines an Nexin 5PE with 7-AAD for determination of live, early, mid, late apoptotic cells, as well as dead cells. The assay uses an annexin 5PE, which evaluates phosphatidylserine exposure, as well as 7-AAD, which allows for assessment of cells whose membrane are no longer intact. An example staining is shown in the dot plot here. Additionally, we show a dose-response curve using the Nexin assay on Jurkat cells that have been treated with styrosporine with different concentrations. The graph clearly shows that the annexin 5 response increases with the higher inducer concentration, as well as higher percentage of cells showing cellular death. One assay that takes things a step further is our MitoDamage assay, which we'll go into a little bit more detail in the next slide, that evaluates, in addition to annexin 5 and 7-AAD populations, the mitochondrial membrane potential. This assay shows clear separation for these very critical aspects of cellular health. The last assay I want to highlight right here is our Guava Cell Cycle assay, which looks at DNA content for your cells. The histogram plot shows an example staining for our cell cycle assay, which uses PI to assess the DNA content. Additionally, the bar graph shows an example on Jurkat cells and PC3 cells that were left untreated or treated for cellular arrest. One final area I would like to show today is related to mechanism of action studies during drug discovery, development, and deployment. Here we show an example of the MitoDamage assay that combines 3 cellular health markers to assess changes in mitochondrial membrane potential, apoptosis, or cellular death. This example deploys all 3 cell health markers simultaneously. Mitosense red detects changes in mitochondrial membrane potential and is shown as a downward shift in fluorescence, indicating a loss of the membrane potential. Apoptosis is detected using annexin 5, which indicates phosphatidylserine exposure. Lastly, cellular death is detected using 7-AAD staining, where there's a loss of membrane integrity. The dot plots shown here show example stainings for the assay on Jurkats. Clear separation for each population is seen. The two bar graphs show dose responses for either etoposide, a known topoisomerase inhibitor, or styrsporine, a known protein kinase inhibitor, and the corresponding population percentages, with depolarized populations shown in red, apoptotic populations shown in yellow, and cellular death populations shown in blue. The bar graph is shown for both compound depolarization and apoptosis. See similar percentages, but little cellular death was seen. Additionally, at this time point, styrsporine shows a much larger shift in depolarization and apoptosis when compared to etoposide. Therefore, in addition to counting and immune cell subpopulation identification, the Guava platforms can be used to evaluate and assess drug mechanism of actions and effects. Thank you. Now I'll turn it over to Stephanie.
Next, we'll talk about the Amnis CellStream, a camera-based flow cytometer which uses time delay integration (CCD) technology. The CellStream system is a benchtop camera-based flow cytometer built on the Amnis technology similar to the ImageStream. An image gallery is used to visualize objects in flow. The CellStream is a high-throughput, compact system with a small footprint, and it allows for modular and flexible upgrades. The high sensitivity of the flow cytometer allows for traditional flow assays as well as small particle experimentation. The CellStream can be used for traditional assays such as immunophenotyping. In this particular study, we performed a 16-color immunophenotyping of major lymphocytes and myeloid subsets in human PBMCs and whole blood. As you can see, we easily resolve all expected populations, as well as populations identified by dim markers such as T-cell subsets. The T-cells were characterized into distinct subsets including naive, effector, effector memory, central memory, activated, and regulatory T-cells. In this study, we use the CellStream to monitor the phosphorylation of signal transducer and activator of transcription, or STAT proteins, which are transcription factors that migrate to the nucleus upon phosphorylation and alter gene expression. STAT proteins are known to be stimulated by certain cytokines, such as interleukins. STATs 3, 5, and 6, which have been associated with prognosis in cancer and other malignancies, were simultaneously monitored in CD4 positive T cells in these experiments. Whole blood was stimulated with interleukins 2, 4, and 6. Red blood cells were lysed and the remaining leukocytes were analyzed with antibodies against phosphorylated STAT proteins, specifically phosphorylated STAT 3, 5, and 6. The gating strategy is shown here for identifying CD4 positive cells. The Phosphoflow results show that IL-4 stimulated STAT6, as in the top panel, and you can see a shift in fluorescence outlined by the box. IL-2 stimulated STAT5, as seen in the second row, and IL-6 stimulated STAT3 in the third row. On the bottom part of this slide, you can see a mix of interleukins phosphorylated all the STATs we investigated. The unstimulated blood is shown in gray, and this is the negative control. Next, we'll shift gears and talk about extracellular vesicles, or EVs. EVs are membrane-derived structures that include exosomes, microvesicles, and apoptotic bodies. Exosomes range from about 30 nanometers to 100 nanometers. Exosomes have been shown to transfer molecules between cells and have the potential to transfer signals between cells. Exosomes are released under normal physiological conditions. However, exosomes are also believed to serve as mediators in the pathogenesis of neurological, vascular, hematological, and autoimmune diseases, as well as cancer. Quantifying and characterizing EVs is challenging due to their small size. High magnification microscopy such as EM can be used to analyze EVs, However, it's very low throughput. Additionally, PMT-based flow cytometers have a difficult time detecting EVs since they are not highly sensitive. Imaging flow cytometers that use a charge-coupled device or CCD camera can detect EVs, such as the CellStream. In order to immunophenotype EVs, we follow this workflow using red blood cells, or RBCs, and platelet-derived EVs. The EVs are obtained from Cellular Biosciences. The RBCs and platelets were washed and treated with a calcium ionophore to induce vesiculation. The vesicles were then isolated via centrifugation. The resulting EVs were labeled with either an anti-CD235 antibody with PE or a CD41 antibody with APC. The labeled EV samples were serial diluted as shown here. Samples were then acquired on the CellStream for 3 minutes per sample with 100% laser power. All proper controls were run, including antibody only, buffer only, and detergent controls. These controls were run exactly as the EV samples. Samples were run in duplicate on 3 CellStream instruments. Shown here in the top row is the gating strategy for identifying the potential EV populations for both the RBC and platelet-derived EVs. The population ranges from about 50 to 200 nanometers with a peak around 100 or 120 nanometers, with the platelet EVs having a larger tail. The bottom row shows the serial dilution data. The 1 to 60 dilution on the far left has a significant amount of coincident events, so that concentration is too high. EV concentration was measured for each of the dilution controls. These dilution controls were used to check for swarming. Swarming occurs when multiple EVs are detected at once or as a swarm, and not as single objects, which is how we want to detect EVs. As you can see here, both sets of dilutions show a linear decrease in the potential EV population using the CellStream for both the PE and the APC labeled EVs. Additionally, the EV mean intensity was measured for the dilution controls. For both PE and APC, the EV mean intensity remained consistent for each dilution. The dilution controls shown here indicate that the potential EV population consists of single objects. In EV research, controls and dilutions are just as important as your samples, since these help determine what is artifact or background noise and what is a true EV. Shown here are the data for the PE positive EV samples and controls and the corresponding serial dilutions for all. As you can see, the average of objects per microliter for the EV samples are significantly different from all of the controls for all dilutions. The low concentration measured for these background controls validates the EV population. Similar to the PE data, the APC positive EVs and corresponding controls show a significant difference, and therefore the EV population is validated here as well. Since the CellStream has such high sensitivity and can easily detect EVs, it is a great system for investigating EV cargo as well. The data shown here are from a study investigating EVs derived from human embryonic kidney cells. These EVs were stained with the Exaglow protein and Exaglow RNA labeling reagents to detect protein and RNA cargo on the EVs. On the left part of this slide, the plots and histograms show the potential EV population positive for protein cargo, colored in green, and RNA cargo, colored in orange. On the top right, dilution series data show that for both protein and RNA positive EVs, there is a linear decrease in signal with extremely low background, validating the labeled EV population. On the bottom right, we see stable mean fluorescence of the positive events for EVs with cargo in the serial dilutions. Protein cargo is shown in green and RNA cargo is shown in orange. The same laser intensities were used for all samples. This indicates the measurement of individual EVs and not swarms of EVs. In summary, the CellStream provides unparalleled flexibility and performance with excellent resolution and detection of target populations, including small particles such as EVs, and it offers high throughput sampling, high adaptability for current and future needs, all in a benchtop system. As you heard from myself, Katie, and Brian, Luminex offers a wide range of flow cytometry products that meet most research needs. As you can see in the table here, we offer a variety of instruments ranging from our cell analyzer to our imaging platforms. Please contact us with any questions about our versatile flow cytometers. Thank you for listening to our presentation.
Thank you, Stephanie, for that great presentation on the CellStream. This is Brian Hall. I'm the product manager for the Amnis Imaging portfolio, and I'd like to wrap up our presentation today by talking about the ImageStream Imaging Flow Cytometer and some of the application used to study cell biology. As you know, cell biology is a general term that refers to a diversity of different types of expertise and applications that might be done, all evolving around cell analysis. Many researchers are interested in looking at bacteria and virus infiltrating cells, or some may be simply interested in measuring protein expression or surface protein expression, while others might be interested in looking at cell-to-cell interactions. There are a number of tools that can be used to look at these different aspects of cell biology, and traditionally this research would be done using flow cytometry. Flow cytometry is a great tool because it collects large number of events. It allows you to look at surface expression and in some cases phosphorylation state of different proteins. And critically, flow cytometry allows you to physically sort cells and do downstream analysis on purified populations of cells. However, it doesn't have any type of morphological information involved with the data set. And so traditionally, if you wanted to look at cell morphology, you would use fluorescent microscopy or confocal microscopy. Microscopy is great because it gives you information on the shape and the size of the cell, but it tends to be less quantitative than flow cytometry because you're looking at smaller cell numbers. The image stream is really uniquely suited to address both of these strengths and allow you to collect images at very high throughput. So the image stream is an imaging flow cytometer. It's essentially collecting tens of thousands of images per second and allows you to collect large image databases that allow you to learn more about your sample. And so it's really combining the strengths of microscopy with the high throughput of flow cytometry. It allows you to make novel discoveries, learn the mechanisms of actions, and essentially get your science right. Now, by combining these two technologies, there are a number of advantages to doing image flow cytometry. One of them is that you can collect data simultaneously. By that, we mean that you can multiplex your experiment so that you could do a apoptosis experiment in conjunction with a immunophenotyping or other type of multiplexed assays. In this example, you can look at the cell surface marker expression and combine that with side scatter or cell health analysis. You can look at where the fluorescence is located. Is it located, is it co-localized, or are the fluorescent markers staining individual subcellular organelles? You can also look at just general marker brightness and quantify the intensity level. And you can also look at colocalization of proteins. So there's a diverse number of ways in which you can multiplex your data, and using all 10 colors as well as the bright field and side scatter information, you can learn a lot about your cell sample. Another advantage to imaging flow cytometry is it allows you to verify your results. And by that, we mean you can look at the histograms and dot plots that are associated with your data set. You can click on those dots and see a picture of the cell. So in this top example, you can not only quantify the number of cells that have 1, 2, or 3 particles phagocytosed, but you can click on these bins and verify that, in this case, this image has 2 particles phagocytosed, and this one has 1, this one has 3. You can also look at marker expression, and by looking at the double positives, you can identify cells that are potentially T cells binding to antigen presenting cells, as in this image, or is the fluorescence overlapping just on the cell surface of one large cell. So it allows you to really understand what's going on physiologically with your samples. And finally, the Amnis team has developed a number of software tools that allow you to really investigate this high parameter data set. Using our regular analysis software ideas, you can look at thousands of different parameters that look at all types of morphological information. However, we've developed new tools such as machine learning and the Amnis AI software that allows you to use deep learning using convolutional neural networks to essentially train the computer to go in and identify unique phenotypes of your specific experiment. So you can customize the analysis based on your individual research. So as you can see, there's really limitless applications that you can use the image stream with. The strength of combining the imagery with the flow cytometry data really allows you to go in and investigate a number of different cell-based assays and look at things like cell signaling, internalization, colocalization, measuring stem cell differentiation, looking at unique cell types such as algae and ocean water, or finding very rare cells such as circulating tumor cells. So there's a number of different applications you can use the image stream for. So to give you a brief overview of how the instrument works, cells are fluorescently labeled in an Eppendorf tube. They are hydrodynamically focused here in a quartz cuvette. We use multiple laser lines to excite that fluorescence. We also use a bright field illuminator to illuminate bright field imagery from behind, so we're essentially collecting multi-mode imagery simultaneously at rates of 10,000 objects per second. Images can be captured at 20, 40, or 60x imaging objective, and that image is projected through a series of Petzval lens sets to what we call the spectral decomposition element. And this filter stack essentially displays the images in spatially discrete regions on a CCD detector. Our CCD camera is operated in time delay integration mode, which effectively allows us to increase the photonic sensitivity and maintain a 0.33 micron per pixel image resolution. So it's very high image quality with very high photonic sensitivity. And again, this is collecting 10 fluorescent channels, 2 bright field channels, and a side scatter image. So it's 12 parameter data that's simultaneously collected on each of your events. This is an example of the type of imagery that it produces. This is a 40x image of THP1 cells labeled with NF-kappa B in green and DRAC5 labeling the nucleus. We capture these images using the camera, but critically these images are pixelated. So in other words, we effectively have not only the total intensity of the cell, but we have thousands of subcellular measurements, so we can look at and effectively map different regions of the cells. So we have an intensity value with an XY coordinate of each of the pixels in the image, and we can then use computer algorithms to go in and interrogate those images to look at unique morphology such as spot formation. These images also maintain spatial registry so that we can overlay these images on top of each other and look at colocalization, look at where the fluorescence is located. Is it in the nucleus or the cytoplasm? And also quantify different morphological aspects of the images. So there's a direct link between the images and the data as they're represented with the dot plots and the histograms. we look through an image gallery here, these images are randomly collected, but we have a region of small singlet cells that we would typically map to a dot plot. Here the singlet cells are in the upper left-hand corner. We also have large singlets, we all mapping here, and we have conjugate cells that map to this lower aspect ratio region. When we look at all 20, 30,000 events in the data file, we can click on a given dot in this image and see its corresponding picture, and vice versa. We can click on the picture and see where it's located in the dot plot. Taking this to the next level, though, we can also look at different surface marker phenotypes. So here are the CD19 cells and the CD3 cells. The T cells are identified there. But looking at the double positive population, we can look at cells that are involved in immune synapse formation. And again, looking at 30,000, 40,000 events, we can characterize and count how many of these cells in this sample have formed immune synapses. Now, the first example that I'd like to talk about is measuring chemotaxis in monocytes after treatment with a chemoattractant MCP1. MCP1 is known to cause monocytes to migrate to the site of inflammation. It's also used in drug development where a patient might have an autoimmune disease, so you can inhibit this pathway and prevent monocytes from causing a disease through self-recognition. In this experiment, we're going to treat these monocytes with MCP1 to induce a change in their overall morphology. And then we're going to surface label the monocytes with CD14, collect 5,000 events on the image stream, and we're going to try and quantify shape change using an algorithm that we call circularity. Circularity is a good example of how you can use quantitative morphology to really characterize the overall shape of your cells. So in the untreated sample, you can see that most of the monocytes are spherical, have a round presentation. If you look after treatment with MCP1, many of the cells have this elongated shape, or some of them are trilobed. So what we want to do is create a feature that can characterize that dramatic change in the overall shape. And again, the feature that we're going to use is called circularity. Circularity, I think, is easy to understand. It's effectively the average radius divided by radial variance. So you could imagine if you have these spokes of a wheel and you have a perfectly round cell, and you line them up from end to tip, when there's very little variation in the total number of radii formed in that circle, you must have a circular shape because all the radii are the same. However, if the cell has an oblong shape, or in this case has 3 lobes, there's going to be a lot of radial variance there. That's essentially how the circularity feature works, is it characterizes how round the cell is versus how oddly shaped it is, and that's also an indication of MCP1, which effectively causes a loss in circularity. Looking at my sample set, you can see that in the untreated sample, most of the cells have this round phenotype. About 9% of the cells have an oblong phenotype. But if I look after treatment with MCP1, you can see that about 60% of the cells are showing a oblong phenotype, meaning that they've responded to MCP1 and are effectively launching an immune response to the site of inflammation. Then the next experiment I'd like to talk about is looking at T-cell antigen-presenting cell conjugate formation and then measuring NF-kappa B translocation to the nucleus as an indication of T-cell activation. NF-kappa B is a transcription factor, so that when it's translocated to the nucleus, you know that these T cells are transcribing genes. And we want to look at this specifically in the T cell to really verify that it's activated when it's bound to the antigen-presenting cell. The problem is that antigen-presenting cells also have NF-kappa B. So what we need to do is isolate the T cell and just look at the NF-kappa B within that specific cell. Now in this experiment, we're using a transgenic mouse that has a TCR that is specific to ovipeptide, such that when we treat the thymus cells from these mice with ovipeptide, we effectively activate the T cells to bind antigen-presenting cells, and launch an immune response. So we're using antigen presenting cells from a different mouse, mixing them together to get T-cell APC cell conjugates, and then we're going to measure the activation as a result of NF-kappa B translocation. So to stain these cells were fixed and permeabilized and stained with NF-kappa B as well as 7-AAD to identify the nucleus. And they had previously been stained with a CD8 to identify the T cells and CD45 to identify the antigen presenting cells. We're going to collect around 30,000 events because this can be a rare event, looking for just those few cells that have formed conjugates. And we're going to try and measure NF-kappa B translocation. Now, the first step in this process is to identify the conjugates. So you can see in the upper left-hand corner of this plot, there are the single cells, and I can verify that region by looking at the images that are in this region up here in the upper left. R3 identifies the cell conjugates, and what we want to make sure is that we're not getting coincident conjugates. So using a CD45 versus CD8 dot plot, we can identify T cell binding to antigen presenting cells, and it's about 80 to 90%. And you can see, in fact, there is one T cell, one APC cell here. So we've identified conjugate formation using a combination of aspect ratio and intensity. The next step is to look at nuclear localization, and to introduce that subject, I need to bring up a subject called a mask. And what a mask does is it's how the computer identifies a region of interest and tells the algorithm sort of where to look for different fluorescence intensities. So in this example here, you can see here's a mask that masks the entire cell. So this would tell me the overall cell size if I wanted to characterize that. I could erode that mask to look at the cytoplasmic area of that cell, but if I subtract the cytoplasmic region from the total cell mask, I can identify a thin mask that identifies the membrane of that cell. So for example, if I just wanted to look at membrane fluorescence, I could use this mask to quantify total intensity there. There are masks that identify the regions in between cells. There are masks that look at dark spots or light spots. And there are masks that look at different components within the image. And so there's a diverse array of masks that you can use. There's approximately 19. And all of these masks, you can use Boolean logic to combine the masks or subtract them. So there's a lot of different ways you can look at subcellular regions. So for this particular experiment, what we want to do is use a mask to identify the antigen presenting cell and the 7-AAD fluorescence. We can then take the APC mask and subtract it from the nuclear mask and identify the nucleus of just the T cell. So this is the 7-AAD and not CD45 mask. I can then use that mask and calculate what we call similarity. And what similarity does is just looks at how much of that fluorescence is inside the nucleus versus inside the cytoplasm. So if I look at a very low dose of ovapeptide, you can see that most of the T cells are not fully activated and that the NF-kappa B resides in the cytoplasm as indicated by the imagery. If I look at a high dose of ovapeptide and look at those images, we get an increase to about 32% response at 50 nanomolar ova. And if I look at the imagery, you can see that the NF-kappa B is in the nucleus of the T cell, but not of the antigen presenting cell. It also has NF-kappa B, but it's located in the cytoplasm. So here, using this masking and different features, we're able to isolate the T cells in this single event and look at the spatial distribution of transcription factor NF-kappa B. It's a really good use of imaging flow cytometry. So using the similarity feature, I can then calculate a EC50 curve. I can do a dose and a time course with that, and look at the temporal interactions of when NF-kappa B is translocating in these activated T cells. So that just is a high-level overview of some of the applications using imaging flow cytometry. There's diverse array of applications that you can use: cell signaling, internalization, cell-cell interaction, spot counting, looking at unique cell types such as parasites or ocean water, looking at rare cells, imaging circulating tumor cells or very rare cells that show up in your sample. So there's a lot of different types of applications that you can use the image stream for that helps you make novel discoveries, learn the mechanism of action, and essentially get the science right. And with that, I'd like to thank you for your time. Please, if you have any questions, type them in and we'll respond to you through email. Thank you very much for your time during this presentation, and I look forward to hearing from you all again in the future. Thank you.